So as you can see, I'm going to be covering who develops motor neuron disease, why, and what we are doing about it. And I talk to you in my capacity as the head of the research lab at the University of Auckland that specialises in motor neuron disease research. So we're the only motor neuron disease specialist researchers in New Zealand, but we collaborate with other researchers across the country, and I'll talk a little bit more about that at the end of my talk. But I welcome any potential researchers who are thinking about motor neuron disease to contact me, as there's always more room for more researchers. So obviously, I'm going to cover these 4 points, but I just want to let you know, I'll take you through what is MND and who gets the disease. And I'll probably spend most of my time talking about why people get MND. It's something that people with lived experience often ask and want to know. And it's something that we as researchers want to understand, because the risk factors for developing MND can help us to understand the pathway by which cells die and look at logical interventions that might be curative. And finally, we'll look at where we're going internationally and nationally with treatments and trials. So, as many of you will know, motor neuron disease is a terminology that we use quite selectively in New Zealand, Australia, and the UK. So in America, mostly the disease is referred to as amyotrophic lateral sclerosis. But the reason we refer to motor neuron disease in this way in New Zealand is that this is a group of diseases. So there's subtypes of motor neuron disease that affect mainly the upper motor neurons, subtypes that affect mainly the lower motor neurons, and of course amyotrophic lateral sclerosis, the most common subtype, affects both the upper and lower motor neurons. So a clinical diagnosis is made usually of either motor neuron disease if you're in New Zealand, or there might be some subclassification of one of these variants of motor neuron disease, depending on the clinical presentation, which we'll talk about on the next slide. But there's a reasonable amount of evidence that despite the clinical differences and the differences in things like rate of progression and the way your disease looks, when we're looking at genetics and understanding what happens in the tissue, many of these diseases at the end of life look the same. So someone who might start off with predominantly upper motor neuron signs or lower motor neuron signs, as their disease develops, they can develop signs in the other set of motor neurons. So a lot of this disease actually collapses down to become amyotrophic lateral sclerosis. You know, if you consider the genetics and neuropathology, they're quite difficult to discriminate. So this is the late onset neurodegenerative disease, and by late onset I mean the 7th or 8th decade, so 60s or 70s. It's a progressive disease, doesn't revert and doesn't tend to halt very much, and it's terminal. It's a fatal disease. So I did say that ALS, or amyotrophic lateral sclerosis, is the most common subtype of motor neuron disease. So it accounts for actually more than 75% of cases. I've already said that it affects both the upper and the lower motor neurons, and we're going to have a little look at what I mean by upper and lower motor neurons in the next couple of slides. And within people who have a diagnosis of amyotrophic lateral sclerosis, most people will have onset in their limbs. So they'll have something like a foot drop as they're walking. They might scuff a toe. They might find that their writing becomes more scratchy. They might find that there's weakness in the limbs. Fewer people have bulbar onset, and that means that the disease starts in the muscles of speech and swallowing, and even fewer have respiratory onset, so the muscles of breathing are affected first. And of course, this is about onset. This means that the disease might start in one region, but amyotrophic lateral sclerosis tends to spread to other regions. There's also a relationship between motor neuron disease, or especially amyotrophic lateral sclerosis, and frontotemporal dementia. So this is a clinical phenotype which is characterized by cognitive decline. People might take on unusual personality traits. They might find that they have repetitive or perseverative thoughts. So FTD, or frontotemporal dementia, is something we look at very carefully if there's a family with some motor neuron disease and some frontotemporal dementia in different individuals. But we actually see, or rather neurologists see, I don't see people in the clinic, this is from reading of the literature, in around 50% of people with motor neuron disease, there's some degree of cognitive decline. And in about 15% of people with motor neuron disease, there'll be frank frontotemporal dementia. So it would be diagnosed in the clinic as such. And although most people with ALS would live for around 3 years, there's a huge variation in this. And for these different subtypes, there's fewer or more people who have a 10-year life expectancy. So this is really can be quite a horrible and scary thing to hear for people with lived experience. But it is a median, and it doesn't account for everyone. So I talked about different upper and lower motor neuron signs, and I won't go over this too much because I'm not a neurologist. I don't look at these features in the clinic, but what your neurologist might be looking for if they were diagnosing you or looking to see whether you have signs of motor neuron disease are spasticity, stiffness, and increased stretch reflexes. So if someone did a hammer tap to the knee, you'd have an increased stretch reflex. Whereas the lower motor neuron signs are more around flaccidity, decreased tone, decreased stretch reflexes, weakness, and atrophy. So that's when there's muscle wasting. There might also be fasciculations, which is where there's quite visible and palpable muscle twitching, or fibrillations, which are more of a kind of a tingle. And the muscle atrophy can be usually is quite significant. You can see this gentleman here, he has a flail arm variant, which means he has amyotrophic lateral sclerosis that mainly affects the upper limbs. And you can see quite severe wasting of his upper arms and also his shoulders. I'm not sure if this is the same person or another person, but they also have another sign of ALS, which is the split hand, so wasting of the musculature of the hand. And this person here has atrophy of the tongue. So when I'm talking about upper motor neurons and lower motor neurons and these different signs, what we're talking about is a set of nerve cells or neurons that extend from the cortex of the brain, the movement or motor area of the brain, and they take a trip down, and they usually cross in the brainstem, and then they will innervate or connect with muscles on the other side of the body. So you can see that you can have this green upper motor neuron here that goes down and makes a connection in the brainstem with a lower motor neuron that innervates the muscles of speech and swallowing. Or we have this blue motor neuron here that crosses over and innervates a neuron that then innervates a muscle of the upper limb, and same with the lower limb. So, for example, the upper motor neuron that's going down here and synapsing in the spinal cord to innervate a muscle of the lower limb, this is an extremely long neuron. This can be a meter long. If you think of your brain down to part of the spinal cord that's around about the level that your limb starts. These are extremely long neurons that have very high metabolic and waste turnover requirements. So there's some special features of motor neurons that we think might be part of the reason they're so vulnerable in this disease. We'll talk about that a little bit in a few slides' time. So who gets this disease? I've got a slide here where I just show a few people who I've picked for various reasons. So I've shown you Lou Gehrig here. He was obviously, most people would have heard of Lou Gehrig, a very famous baseballer in the US. And in fact, in the US, if they don't call motor neuron disease amyotrophic lateral sclerosis, they often call it Lou Gehrig's disease, because he was probably the most famous person to have developed the disease. And there's very interesting kind of statistics around Lou Gehrig's baseball batting average, and kind of tanked at the end of his career as he developed motor neuron disease, or ALS. Here we have my friend, the lovely Claire Riley, who many of you will know. She's the research strategy manager for Motor Neuron Disease New Zealand, and we recently attended a conference together. She's very active as a researcher herself, and is a trained medical doctor who has MND. Stephen Hawking, another very famous example. And I've just chosen this gentleman here. I hope he wouldn't mind, but I picked his image off the internet, and I've chosen him because he looks like he's attending a charity walk, and I just wanted to raise awareness about the motor neuron disease charity walks that are held each year. They're called Walk to Defeat, and they're one of the main fundraisers for Motor Neuron Disease NZ to raise money for care and support of people with MND, and also some funding is often ring-fenced for research. And what I also wanted to say about this slide is, I suppose there are a couple of things that are representative about who gets MND on the slide, and a couple of things that are anomalies. So this gentleman here, he's probably more your average person you might expect would be at risk of MND because he's elderly, he's white, and he's a male. So we know that these three here, Stephen Hawking developed MND in his 20s, I believe, and Lou Gehrig and Claire were also very young when they developed MND. That there are people that develop MND as a younger person, but I've said already that the majority of people will be in their 60s or 70s. Another thing I mentioned is that he is male, so I'll talk in a few slides' time about the fact that males are slightly more likely to have MND than females, and he's white, so I haven't got any people of colour on this slide. Of course, people of colour do have MND. We'll talk about that in a few slides' time as well. But the research suggests that ancestral or ethnic admixture might be protective against MND. And there is a lot of research going on to unpick whether the lower rates of MND in people of colour are around equal access to diagnostics and neurological services, or if it's a genuine protective effect. So this is a disease that's 90% sporadic. So sporadic means out of the blue, unexpected that a person might get MND because there's been no other person with MND in the family. For the other 10% roughly of cases, there will be a family history of disease. Someone else in the family would have had MND or frontotemporal dementia. Internationally, the number of people, or the rate of people who are diagnosed with MND each year is around 1 to 3 people for every 100,000 people. In New Zealand, we think that rate is a bit higher. So my PhD student, Maisie, completed a study in 2018 where it suggested, and I'll talk about that next slide, suggests that our rates are higher. And another paper in that same year that also looked at New Zealand supported our findings. So based on this rough number of 2 to 3 per 100,000 per year and a back of the envelope calculation of 5 million New Zealanders, approximately 100 to 150 New Zealanders develop MND each year. So just drilling down into that a little bit more for New Zealand, using the data that we've collected previously, we did find this higher rate than what is expected internationally, or at least in other countries with similar healthcare systems where the access to diagnostics would be assumed to be equivalent. We found that the rate of male versus female, I say here diagnoses or deaths, when we did this study, we used death certificate data. So we weren't assessing people being newly diagnosed with MND, or even everyone living with MND at that time. We used data from the New Zealand Statistics Department, which told us how many people had MND listed as a cause of death on their death certificate. And we used that to infer how many people would have been diagnosed. Because MND is a terminal illness, there's usually a good relationship between the number of people that die with MND and the number of people that have MND. Few people will die of other causes, although of course some will. We found that the male to female ratio was about 1.2 to 1.5. So for every 1 woman with MND, there'd be around 1.5 men. Most deaths from MND were in 70 to 79-year-olds. We found that the rate of MND in New Zealand Māori was lower than that for the total population, and probably around 2 people per 100,000 per year, but the rate was increasing over time. So we think that there may have been access issues that are narrowing, the gap is narrowing between access to good healthcare and good diagnostics for Māori and the rest of New Zealand. But we're also mindful of the fact that there might be protective factors in Māori, and lifestyle or genetics. And we do work closely with several New Zealand Māori families, and we're very keen to work with and partner with other Māori who have an interest in MND. So another thing we found is that the rate of MND diagnoses or deaths was not increasing above population aging. So what does that mean? That means if we look at this graph here, this is a prediction of what the New Zealand population looks like over various years. So at the time that we did this study, it was already 2018, so the data for 2018 was predicted only. And predictions were made about what the New Zealand population would look like from 2010, 2018, 28, 38, 48. And if you follow the data in the grey line, you can see the proportion of people that are 0 to 5 years old is about 0.07, so 7% of people. And you can follow that line along and see there's a little bit of a hump, but a few more people, the proportion of people that are around 15 years old was a bit higher. Same here, another hump. So the proportion of people around 45 years old was a bit higher. And then if you look in the kind of 70s to 80s age range, fewer people are of that age. But as we look up at predicted population structures through to 2048, the number of people in this age bracket is increasing each decade. So we know that if you have an aging population and you have an age dependent or a disease which has age as a factor, late onset disease, then the older the population gets, the more people are at risk of developing that disease. So we think that population aging is going to be one of the main drivers of more people in New Zealand developing MND. So why do people get MND? I'm going to spend a little bit of time on this section because this is where we do most of our work. And I think there's a lot of potential here for understanding MND more deeply. So I've already said that around 10%, some people say it's more like 5 to 10% of ALS is familial. That means there's a strong family history. But this indicates, because there are 90 to 95% of cases where there is no family history, this indicates that although there might be genetic factors involved, that the genetic factors are either not strong in many people or then not the only factor, might not be present in everyone. So the most widely accepted model of risk in motor neuron disease is described by this reasonably complicated graph, which I'll take some time to walk you through. So it's called the multi-step hypothesis. And along the x-axis, or this line here, is time. And along this axis here, there are several different things being measured on the y-axis. And we'll talk through first the screen, the molecular damage, and then we can also look at physical health and decline in things like clinical stage, physical health, and functional stage. So if a person starts their life with no predisposition, no genetic predisposition for motor neuron disease, they will require 6 independent molecular damage events to occur over their lifetime. And some of those possible molecular damage events are listed here, and I'll talk some more about others on the next slide. And if a person has kind of this unfortunate hand of cards, at some point over their lifetime, they're exposed to 6 individual factors that are MND risk factors that have enough effect to take them up to this kind of threshold, then this is the threshold for development of MND, after which time they will have onset of disease and decline in physical health, along with decline in these measures of physical health. There are other people who have some genetic predisposition. They might have some genes that are risk factor genes, or genes that changes in these compared to others have a small effect on risk. So they might only need 5 molecular damage events to happen during their life, or 4. And then there are others where they have a high predisposition to developing MND because they have inherited a gene with a strong effect. So these people will have a family history of MND. They'll have many other individuals with MND in the family. And when they start life, they already have accrued a lot of molecular damage from that genetic change that they've inherited. And they might need only 2 or 3 additional molecular damage events to happen during their life. So they might have onset earlier in life, or they are certainly more likely to meet that threshold and to develop MND, because they only need those 3 more steps. So the molecular damage events that I've been talking about can fall into either environmental or genetic categories. But this work is very, very hard to do. And I know a lot of people have questions about this. They are wondering, why did my loved one, or why did I develop MND? Is it something that I did? There is a lot of the blame game that goes on, which is absolutely understandable. You want to know if what's happened was avoidable, and if something can be done to help others, if these risk factors are still at play. But this work is very hard to do because, as I said, the accrual of molecular damage events is basically happening across the lifespan of the individual. So we don't know how long before developing MND the exposure of interest happened, and we don't know what the exposures are that have the most effect on risk. So there have been some environmental factors that have been reported internationally, but I put question marks over all of these because for almost all of them, for every study that says that there is an increased risk of MND with exposure to this factor, there will be another study that says that there is no increased risk, or even that the factor is protective in some cases. So these are military deployment, electromagnetic fields, agricultural chemicals, head injuries, smoking, and professional football. And I'll just point out that one of the reasons, aside from the absolute plethora of things we're exposed to over our whole lifetime, and the difficulty in kind of pinpointing the time that is of interest, many of these studies that find an association of factors with MND are very small. So one person developing MND in the control group versus the, say, the military deployment group, one way or the other, that can skew the statistics quite a lot because of the small size of the study overall. So that can have a big effect. Also, most of these studies, rather than starting with a group of people, none of whom have MND, and looking at their exposures and following them to see who develops MND, those studies would require tens or hundreds of thousands of people. What is mostly done is groups of people who already have MND and some controls are looked at and asked to recall things that they've been exposed to. And sometimes there can be bias in that approach because people might have already heard that there's an association with head injuries or agrochemicals, so they might be more likely to recall that as something they've been exposed to. Also, if there's only a single method of identifying cases, so only questionnaire or only sending out an online answer sheet, then you can have bias in terms of the people that are able to fill those out. If you were looking for footballers who developed MND, for example, you sent out a questionnaire to football players, people who had developed MND might be more likely to respond. In New Zealand, we have also had many environmental factors that have either been reported in the news or described in scientific papers. But again, I think we'll discuss that small sample sizes and methodology can unpick the confidence in these studies or these reports. So one of them that received a lot of press a few years ago, probably 10 or 15 years ago now, was the log fumigant methyl bromide. So this is used in the Port of Nelson or at least it was, not sure if it's still being used, to fumigate logs that were to be used for wood. And at the time, I think it was around 2002 to 2005 or so, there was what the public had considered to be a cluster of people who developed MND. And it was requested that the medical examiner, Ed Kiddle, looked into this cluster to determine whether the group of people who developed MND in this small area, whether they had more exposure or it was due to them working with this methyl bromide fumigant. So his conclusion was that it was a random occurrence or chance that the cluster occurred, and clusters can and do occur by chance. So and part of his report, and you can find it online, stated that the use of methyl bromide in the Port Nelson area was not more than other areas internationally. And actually some of the people who developed MND were not actually port workers, and they were unlikely to have had exposure to the fumigant that was significant. And despite that finding, there's been a continued reporting about methyl bromide. This was in 2018, there was an article. So I think we don't have good evidence to suggest that methyl bromide is a contributor to risk, but then these risk factors are very, very hard to verify and to substantiate. So it could be possible that methyl bromide, along with many other things, could be a risk factor. Another one that reached the news is painted apple moth spray that was sprayed in West Auckland in the early 2000s. There were a number of people saying that they had various symptoms from the spraying, and MND was one of the things that people felt that there might have been a relationship with, but at the time MND NZ came out and said there was no statistical cluster. So again, as much as it's very important for people to report when they feel that there are more people or that there's a cluster developing, these situations have to be looked at statistically with a view to understanding what is the overall population in that area. Are people migrating into the area to access health services that have MND that wouldn't normally be there? What is the exposure for the people that developed MND? Was it more than others? And did people with more exposure, were they more likely to develop MND? And if this kind of proper statistical analysis is not done, you can have reporting that scares people essentially unnecessarily. So some other things that have been reported by scientific studies rather than just in the newspaper, and this is by a group at the Centre for Public Health Research in Massey. They've reported a number of different potential risk factors. Again, their study was larger than many, but still quite small. So whether these factors increase risk greatly for MND is still unclear. And I'm just going to remind us of this, this issue in finding environmental factors, and I'll take you on to a comment by an expert.
Are there things in the environment that are causing this in people?
Well, the first thing to say is that we already know some of those same genes that cause familial ALS also cause apparently sporadic ALS. So between 15 and 20% of people with no obvious family history can be found to have a genetic variation in one of those genes. And in most, but not all of those cases, there's strong evidence that that's probably contributing to the cause. Finding environmental risk factors is much more tricky because with the genome, although it's vast, it's a finite thing and we know what we're testing. With the environment, it could be absolutely anything at any point in the person's life or a combination of those things. And so it becomes extremely difficult to try and disentangle that. There are various techniques that can be used to try and attempt that. Jinsey's mentioned some of them, which is basically epidemiology studies, but those are dependent on the recall of the person who's being asked. And that, particularly for distant things, is not good. And for example, smoking, something like 25 different studies have been done looking at whether smoking is a risk factor for ALS. And about half of them say it is, and about half of them say it isn't. And for example, we've done a very large study called the Mendelian randomization study, where you look to see if there's a causal relationship between smoking and ALS, and we found none at all. And so that tells you the difficulty in finding these environmental risk factors.
So that was Amal Al-Chalabi at King's College in London. And he talked a little bit about genetic risk factors, which I'll move on to now. And he talked about the fact that some of the same genes are found in people with familial MND as are found in people with sporadic MND. So I'll go over that in a little bit of detail. So we know so far that there are about 40 different genes, any one of which can cause MND if it has an error in it. The most commonly found gene to cause MND is one called C9orf72, not very catchy. But you can see this in green here. In people with familial MND, it makes up about a third of those cases, and in sporadic disease, it might be probably 7 or 8%. We have another few genes here, and I've highlighted SOD1 as a particular example because we've also found that in New Zealand. We'll see that on the next slide. But one thing I wanted to highlight is that, even though we know for international cases, around 70% of those cases we have a genetic answer for, and far fewer of those sporadic cases we have an answer for. When we do have a genetic answer for sporadic cases, it's the same genes. And because this overall group of people, the sporadic MND cases make up 90% of all MND cases. So this circle actually comprises a vast number of people compared to this circle. So this green portion of people with sporadic disease who have the C9orf72 gene mutation, the number of people in here is actually greater overall than the number of people in here. So while trials and studies look to recruit people with familial MND with C9orf72 gene mutations to test whether therapies might work for them, actually there are far more people in this category. So it's very important that we conduct genetic screening on people with sporadic MND as well, because they might be eligible for, and they might benefit from, these treatments that are directed towards that gene. So what do we know about these same genes in New Zealand? Again, I've split it into familial and sporadic MND, and we haven't done quite as well as those international studies in kind of ticking off causes in people, but we have found, again, around about a third of people, numbers are very low as yet, but we have probably another 50 or so people who we are expecting results on soon. Around a third have the C9orf72 gene mutation, and a small proportion of them, of sporadics, have the same, and SOD1 is probably our second most common genetic cause. What we also see is these genetic variants of uncertain significance. So these are differences in the genome compared to most other people that we don't really know what they mean. And I'll talk about that on the next slide. There's another gene here that is a risk factor gene that can increase the rate of progression of MND. We're very interested in this, but it's also found in people that don't have MND. So it has to be taken with caution. So I said, we sometimes find differences in people, in their genes from others, and we don't know if they cause MND or if they're just normal variation. So I'm just going to take you back to sixth form or year 12 biology and remind you that your DNA is a bit like a recipe book, and a gene is an individual recipe. And the instructions here can be photocopied, a little copy called mRNA, and they'll help you make a beautiful cake. I say cake because I'm a cake fan. But if there's a change in a single word in that recipe, then the photocopy will obviously have the corresponding change, and you might end up with quite a different cake. And the cake might be a different shape, it might not do its job, might not be delicious, there might be a lot more, a lot less cake, or it might be in the wrong place. There's also some genetic changes that don't change the shape or function or amount or location of a protein. Or even if they do, the change may or may not be damaging. So we have the job of, even once we've interrogated the genome of people with MND, of understanding whether a difference from other people who don't have MND is a mutation that causes their MND, or if it's just variation, just normal differences between people, that's benign. So how do we test that? And this is where my lab does quite a lot of our work, drilling down, trying to understand the consequences of having a genetic difference from others. And we work in particular with a family who have a gene difference in a gene called ubiquilin-2. And we have been very lucky to work with brain tissue from deceased donors from this family. And in this family, this is a piece of the brain that has been stained or labeled with lots of different antibodies, and we can put them in different colors. They pick out different proteins in the tissue and put them in different colors for us to look at. So we can see things like blood vessels, these guys here, things like neurons, and this is the nucleus, the kind of instruction or library area of the cell. If we zoom into a particular part of the brain, we can see the ubiquilin-2 protein, which is the one whose instructions are contained in this ubiquilin-2 gene. Instead of being nice and smooth as they are in controls that don't have MND or don't have this gene change, they form these clumps. And they're recognized by another protein, which is a bit like a waste disposal tag or a come eat me signal. So we know that for this gene change that we see in this family, this biological evidence tells us that it's a mutation and not just normal variation between people. We can also test that by looking at other people with changes in that same gene, but in different parts of the gene. So we're comparing our family, which are the two people in the middle. They've got a gene change at this particular location. Yes, a gene change. We can compare them with other people with ubiquitol 2 gene changes, this person here and this person here, and all of them have clumps. Ubiquitol 2 clumps here, 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 and here. So it tells us that a gene change in this part of the gene is just as damaging as a gene change in this part of the gene or this part of the gene. And we also always have to compare our brain donor tissue back to that from controls, and also from people that don't have any, have MND but don't have a change in this particular gene. And we see no protein clumps in either of those groups. And might I take this moment just to acknowledge the brain donors that donate, people with MND, that their families and the people affected by MND donate to our brain bank, and also people who don't have a neurological disease. This control tissue is just as important for us to understand why things go wrong and what going wrong looks like. So another thing that we do through a collaboration with a group in Australia is looking at the genome of people with motor neuron disease and looking for chunks of the genome or the genes that are shared between people in New Zealand who have MND and people in other countries that have MND that suggest that they're distantly related. So our collaborators have worked with about 4,000 samples from a number of different countries, and from our cases, they found one sample where the person is very distantly related. It's a 7th degree relationship, so the last common ancestor was a great, or even later grandparent, and they're distantly related to this person, and this person is related to all these other people. All these dots represent people. This cluster of people all have the exact same gene change that we know causes MND. So this is evidence that the same gene error or mutation can be inherited and travel basically across countries, continents. But this looking at distantly related people can help us. For example, if we had a gene change in this person that was shared by all these other people and they all have MND, it's another way that we can understand whether that gene change is damaging or if it's just normal variation. So I'm going to just take a little bit more time. So I'm running a little bit long, but I've only got a few more slides. I think this is very important to talk about what we in New Zealand and what international research is doing about MND. So it's a busy slide, but I'll take you through. The treatments that are already available and approved for use in people with MND are firstly, and I've highlighted in green the ones that are available in New Zealand. So non-invasive ventilation, this is usually BiPAP. This is a treatment people can use assisted breathing, and it's particularly used overnight, that can be used during the day. It's known to extend life, and many say that this is the treatment that extends life by the most of any, far more than any of the neuroprotective treatments. I don't have the numbers on how long the life extension is, but your neurologist should have that if you ask them. It's known to also improve quality of life. Riluzole or Rilutech is a pharmac-funded drug that's available in New Zealand. And this, in the initial study, it was shown to extend life by around 3 months, but that was across a 12-month study. So the advice now is that it extends life by about 38%. So a person that was expected to have a lifespan with MND of 10 years would have a longer extension of life on Riluzole than someone who had a shorter expected lifespan. And edaravone is the other FDA-approved treatment for MND. It's not available in New Zealand as far as I know, and the improvement in function from the trial looks quite small, and it's really been shown to be effective in just a small subset of patients. The trials that are underway internationally, and which we in New Zealand are passionately advocating for New Zealanders to be able to have places on, or for us to be able to run trial sites in New Zealand, are listed here. There are some gene therapies against SOD1 and C9orf72, so those are the two most common genes. There's also one for that risk factor gene I talked about, which can increase the rate of progression, but is also found in controls. So this lithium treatment has been shown to restore people to a normal rate of progression with MND. It's not a cure or it won't halt disease, but it should restore a normal rate of progression with respect to other people with MND who don't have that genetic variant. There are a number of other things that are being trialed with various success. I just would draw your attention to Neuron, which is a stem cell treatment that takes a participant's own stem cells and reprograms them or genetically modifies them to produce some growth factors and neurotrophic factors. So this has been trialed in clinical trials and has been proven to be safe. I don't think there's any efficacy data out that may be available now. If you want information on any of these or other clinical trials in ALS or MND, go to this website. This is the US trials, but they also cover trials that have sites in other countries. But I would caution you about stem cell treatment. If you're looking to find a stem cell provider in New Zealand, these stem cells are very special stem cells that have been modified to do something special. They're not your everyday stem cells. So to take data from these and extrapolate that to infer usefulness of other stem cell treatments is not a good idea. If you are thinking about taking something that's not been approved, or I mean, I would at least caution you to only look at things that have been proven to be safe. The ALS Untangled website is really great for a group of clinicians come together and do a review on different treatments that have been suggested by the ALS community, people asking, is it safe to take X, Y, and Z? And the ALS clinical expert community will come together and review that. And I highly recommend that site. So why don't we have trials in New Zealand? And I want you to know that we are trying so very hard to get this to happen. When I say we, it's kind of the royal we, because I'm only very peripherally involved. Often I'll see something and let people know about it, but I don't have any clinical usefulness or ability to run a study myself. But I can communicate to you why we don't have these trials. And part of the reason is that we have few people in New Zealand with MND. And if our cohort of people with MND are not well characterized in terms of their genetics or their rate of progression, things like these, it makes it very difficult to know if we have the right number of people who would be eligible for a trial site to be started in New Zealand. So please do consider joining the registry and joining our genetic study that we run at the University of Auckland. The more people that we know about with MND, obviously the harder we can advocate for running these trials in New Zealand. We're also very distant from other trial sites. You know, the main trial sites are overseas largely. People are quite geographically spread in New Zealand, and we don't have centralized clinical care. The cost is high and we don't have necessarily good funding to cover these trials. And also it's a matter of our neurology and support staff being basically at capacity already. So it's difficult for them to take on new things. Money could help most of these things. More people being involved in research always helps. And any advocacy that you would like to participate in, you would be very well accepted and well appreciated by MND NZ if you get in touch. So on my final slide, I just wanted to bring hope, I think, I hope, to and show you what the infrastructure is that's involved in MND research in New Zealand and how this might change the situation from the previous slide where we don't have any clinical trials. So we have in New Zealand an absolutely wonderful advocacy and support organisation in MND NZ. And many of you will interact with them already. If you don't and you have MND, then you should avail yourself of their services. And they also offer a research oversight, because MND NZ are a funder of various other parts of the infrastructure. They have good oversight as to what's happening kind of across the board, and they're able to to drive that in some ways. So one of the things they fund is the MND patient registry. This helps to recruit people with MND into research that they're interested in. You can sign yourself up to the registry and be offered a place on a research study. Eventually, when we have trials in New Zealand, you'll be offered a place on a trial if you're eligible, and it's up to you to decide whether you'd like to participate or not. You're under no obligation. There's also a research network. So I run this, and this is a researcher-facing network of people who are all doing MND research and like to work together for best efficiency. I've talked already about the national genetic study that we're running, and I hope that anyone who's not involved in that study would consider now being involved. We are always open to enrolling more people. And of course, I need to acknowledge the Human Brain Bank at the Centre for Brain Research, who we work very closely with to conduct our human brain studies, and the donors and families that are involved in supporting tissue donations we can't do our work without them. So I would say that this infrastructure that we've built so far is really, we've now got our the foundation stones in place upon which we can build clinical trials. So to be trial ready, people with MND should be mobilized. We should know who they are and where they are. That's the job of the registry. They should be well monitored. So our clinics are talking about best practice guidelines for looking after people with MND. They should be characterized genetically. That's what our genetic study does. And we're advocating for more clinical genetics, which is much quicker than our research genetics. And people should be able to donate samples if they so choose to. So I hope that has given you a whistle-stop tour of what we know about MND and what we don't know about MND and where things are going. And I must thank, this is all an absolutely enormous collaborative effort between my lab and many other labs in New Zealand and across the world. And in particular, we work very closely with neurologist Richard Roxburgh and Dipna Mulroy, who's the research network coordinator and also the patient registry curator. And Miriam and Christina, some of you may have met, and Harry now, who consent people to our genetic study. And of course, our very generous funders. We could not do this work without you. So thank you very much, and I'll hand over back to Sarah Jane to cover some questions.